Hello everyone, welcome you all to BIOSKUL. This is Gayatri. In today's session, we are going to discuss about regulation of gene expression in prokaryotes. This is the part 2. In part 1, we have discussed negative control of the Lac Operon. You will get the link in the description box of this video down below. In today's session, we are going to focus on positive control of Lac Operon. So let's proceed and know how the Lac Operon it is positively regulated. You know, Lac Operon, it is consists of a group of genes which is regulated by a single promoter. Lac Operon, it is controlled both by the positive way and also it is controlled negatively. And that depends on two different proteins. In negative regulation of Lac Operon, a protein, that protein is known as the repressor protein, it prevents the binding of RNA polymerase to promoter, thereby it inhibits the transcription. That's why it is known as the negative control of Lac Operon. In positive control or positive regulation of Lac Operon, the protein which is responsible is known as the catabolite activator protein or CAP protein. This protein, it helps in binding of RNA polymerase to the promoter, thereby it enhances the rate of transcription. Now we will know in details about the catabolite activator protein. The catabolite activator protein or the CAP protein, it binds to a site. The site is known as the CAP site or catabolite activator protein site. This site, it is present just upstream to the promoter region. Catabolite activator site, it is also known as the catabolite activator protein binding site, that is the CBS, or we can also call it as the CRP. CRP is the cyclic AMP receptor protein. Okay, so this CAP protein is bind to the CAP site. It is a dimeric protein. Dimeric protein means it has the two binding site. One binding site for the cyclic AMP and another binding site for the DNA. So when the CAP protein it binds with the cyclic AMP, there its affinity for DNA increases. The bound CAP protein with cyclic AMP, it promotes the transcription by binding the RNA polymerase to the promoter efficiently. Because the Lac Operon, it has the weak promoter. So the RNA polymerase alone, it cannot efficiently bind to the promoter region and it will make a very few transcripts. But when the CAP protein, it binds to the DNA, it enhances the binding of RNA polymerase to the promoter region efficiently and help in efficient transcription. Now the question is whether the CAP protein it is always active? No, it is not always active. It is regulated by a molecule that is known as the cyclic AMP. And this cyclic AMP which in turn regulated by the glucose. So when the cyclic AMP it get expressed, cyclic AMP it get expressed when the glucose levels are low. So CAP protein it is not always active. It depends on the cyclic AMP and the cyclic AMP expression that in turn depends on the glucose concentration. So when the glucose concentration is low, at that time cyclic AMP expressed more and the cyclic AMP it will bind to the catabolite activator protein or CAP. So when the CAP it binds to the cyclic AMP, then it will help in the binding of RNA polymerase to the promoter efficiently. So now the cyclic AMP its regulation depends on the concentration of glucose. When the glucose concentration is high, at that time the high concentration of glucose it inhibits the enzyme that is adenylyl cyclase, and this enzyme it helps in the conversion of ATP to cyclic AMP. That's why when the glucose concentration it will be low, then it cannot inhibit the adenyl cyclase. So adenyl cyclase it will be active and that will produce the cyclic AMP. Okay. Now we'll see when the glucose concentration is low, that means the cyclic AMP concentration it will increase or decrease. So when the glucose concentration is low, then adenyl cyclase it will active and it will convert the ATP to cyclic AMP. Now the cyclic AMP concentration is high. So the cyclic AMP now it can bind to the catabolite activator protein CAP. When the CAP it binds to the cyclic AMP, they together it will bind to the site. That site is known as the catabolite activator protein site, that is the CAP site. Okay. So when it binds to the CAP site, then at that time it will enhance the binding of RNA polymerase to the promoter region. Okay, it will now the RNA polymerase it will bind to the promoter efficiently, so that that results in the high rate of transcription. Okay. Next we will see if the glucose concentration is high. Glucose concentration is high, then adenyl cyclase it will be inhibited. If the adenyl cyclase it is inhibited, then the cyclic AMP concentration it will decrease. If there is no cyclic AMP, then CAP protein it will in inactive state, and the inactive CAP protein it cannot bind to the CAP site. So CAP site it is blank. Now the RNA polymerase it will bind to the promoter of Lac Operon. Is the promoter of Lac Operon it is a weak promoter, so RNA polymerase it cannot bind efficiently to the promoter and it will produce few transcripts. That's why that results in the low rate of transcription. Hope now you have got a clear idea about how positively Lac Operon is regulated. If you like this video, then please do share with your friends and don't forget to subscribe my channel. Still, if you have any confusion regarding regulation of gene expression in prokaryotes, then put a comment in the comment section down below. I will get back to you as soon as possible. Thank you so much for watching this video. Bye bye. See you in my next video.